I'm Daniel Levine, and this is the Bio Report. The traditional approach to treating autoimmune disease has relied on ways to suppress the immune system. COUR Pharmaceuticals is developing first-in-class therapies that instead seek to reprogram the immune system to create antigen-specific tolerance. We spoke to COUR CEO John Puisis and COUR Vice President of Research Adam Elhofy about how the company's immune-modifying nanoparticles work, how the approach preserves the immune response, and its partnership with Takeda focused on celiac disease. John, Adam, thanks for joining us.
Thank you, Danny. Totally appreciate it.
Thank you.
We're going to talk about autoimmune disease, COUR Pharmaceuticals, and its efforts to develop first-in-class therapies designed to reprogram the immune system. Well, let's start with some basic. What goes awry when the immune system in the case of an autoimmune condition?
Yeah, I'm going to jump in. This is John, and then I'll have Adam chime in as a follow-up. But the immune system is at pretty much basic levels, very quiescent, very regulatory. And somehow, the immune system is built to take out foreign products like bacteria or viruses, and it's a surveillance system. And for reasons that all scientists don't have the exact reason why, somehow the immune system starts to look at a tissue and recognize it as foreign. So let's take women who have primary biliary cholangitis. It's a liver disease. Somehow, and it happens in women of middle age, all of a sudden, T cells start to recognize the bile ducts within the liver as being foreign, and they start attacking the tissue, which creates inflammation, and eventually it creates sclerosis, which eventually leads to liver failure. So the immune system, which is a very protective system for us and our bodies, actually can be very deleterious and dangerous by these triggers, and we call them mimetopes, if you will. There's just some trigger event that occurs. Some of it has to do with the lineage of the people, but sometimes it just could happen out of the blue, where the immune system takes its own action at attacking a certain tissue within the body.
Yeah, I'll take it just a little bit further. All of us have cells that are circulating that are specific to self. And essentially, to extend a little bit further on what John says, is that essentially, in some cases, those cells that are circulating to self become activated. And in most of us, our immune system is able to regulate that response such that it doesn't become pathologic. But in people with autoimmune diseases, those T cells do become pathologic, and it starts a positive feedback loop where once that inflammation occurs in the tissue, essentially it then becomes almost a self-fulfilling thing where now there's inflammation. It says that it should progress on and that it is a good response to have, and then the autoimmune disease perpetuates. But in most of us, we find a way to control it, and that control is called tolerance.
As I think about the way these conditions are treated, they generally involve efforts to suppress the immune system either through the use of steroids or through more targeted approaches. How effective have these been?
Well, Danny, you're talking to people who have changed their religion away from immune suppression and more towards immune tolerance. So, if you watch the morning news, there's just a series of commercials related to all types of autoimmune diseases and inflammatory diseases. And what those products do is just suppress the immune system. So you still have the disease, but some of the symptoms are relieved, but at a great cost, and that's a compromised immune system. My cousin, for example, he has rheumatoid arthritis, can barely get up in the morning and comb his hair, has a hard time moving, joints are always achy. So he went on a series of immune suppressants for decades, and he developed other issues related to being immune compromised. So the industry for the last 20 years has made hundreds of billions of dollars, has been very successful at productizing these immune suppressants. And what we want to do is bring something very revolutionary to people, where we actually shut down that autoimmunity completely, and people don't have side effects from that.
Well, how problematic are side effects with regard to these therapies?
They could be significant, from having occasional infections to having serious infections to gaining cancer. I'll tell you again, my cousin who has rheumatoid arthritis, I won't say the product that he was on, but he ended up with sepsis and he almost died. And the sepsis was a result of being on immune suppressants for a very long time.
And some of the other drugs that are prescribed are steroids, and steroids, typically the way that the treatment approach is to give the patient steroids until they get something that's called Cushing's disease, which means that essentially they've maxed out on how much steroids they can take, and then they back off. And Cushing's disease is somewhat dangerous, but then the side effects of being on steroids for prolonged periods of time can include bone pain, amongst other things. So it's not trivial, the side effects that occur when you're on some of these prescriptions for a prolonged period of time.
COUR has developed a platform, immune-modifying nanoparticles, to treat the root cause of immune diseases. This trains the immune system to tolerate specific antigens. What does a COUR therapy consist of, and how do you determine the appropriate antigen for a disease?
We actually we do a lot of research. We look at things that have been published. We looked at things that have been published and looked at for decades. And we go to diseases where we know the culprit antigens. And it's been widely accepted by the medical community, by the science community, well published. And so we know what antigens that are the target antigens. And then we run mouse models and other models, and we actually see the effect before we go into humans. So it's pretty well publicized, a lot of these antigens, because in every field, there are doctors who are looking at trying to cure the disease. So they get the first line of defense for them is to look at what are the known antigens. How do I shut them down? How do I change them so that the person could have a better life?
And in terms of the therapy, though, are you just delivering the antigen in a nanoparticle, or is it packaged with anything else?
It's the nanoparticle is not a carrier. So let me try to frame this, and Adam will probably chime in. Our nanoparticle is about 500 nanometers, and what that means is it looks like a cell, and it goes into the bloodstream. It has the antigen inside there, but the whole particle, plus it has a negative charge to relate to the immune system. And that particle looks and gets observed by the immune system as debris, cellular debris. And we call it apoptotic debris, meaning dying cells. And what the immune system will do is we'll pick up those dying cells, but there are particles, we'll bring it to the spleen and liver. And in the spleen and liver, there's a lot of antigen presentation through antigen presenting cells. And what's cool about this is once it's in the spleen and liver, the antigen presenting cells chew up the particle and chew up the antigen, and they recode they being the immune system, recodes the body to say, don't go after these antigens anymore. They're good. They're part of the body. They're part of cellular debris. And it turned out to be quite fascinating. It's been a work of a number of scientists over 20, 30 years. And is really a great discovery.
And they're delivered systemically?
Yeah, we do intravenous, because your bloodstream is where you have a lot of apoptotic debris. It's the best place to have the immune system kind of cleaning up the system, if you will, and bringing it to the spleen and liver. That's the most efficient way of delivering these particles to where they need to go.
So, I mean, let's differentiate it a little bit from like liposomes that are delivered systemically. Because of their size, there is systemic distribution that's potential with liposomes. Because of their size, they'll end up being taken up by a variety of different cell types, whereas our particles, with the size that they are, they'll predominantly be located in the spleen and liver. So even though the particles are delivered IV, they are not systemically distributed. They end up in the spleen and liver. And so there is a kind of targeted approach there. And then the cells that are capable of taking up the particles are limited to antigen presenting cells, because those are the only ones that are able to take up particles that are greater than 300 nanometers in size. And so there's specific targeting to those cells. And then further, the part that is critical is that there needs to be a tolerogenic signal, and it needs to be a tolerogenic signal that's in the context of the antigen. And so the tolerogenic signal comes from the negative charge on the particle. So now you have particles that are essentially destined towards the liver, delivering a negative charge to the antigen presenting cell. And then the antigen presenting cell has a tolerogenic signal. It then takes up that particle or phagocytoses that particle. The antigens are released, and then now that antigen presenting cell can represent those antigens in the context of tolerance. And that's where the reprogramming occurs. So it goes from that antigen in the prior scenario in autoimmunity being presented in the context of activation. Now, after delivery of the particle, there's reprogramming, and now that antigen is being presented in the context of tolerance. And that's where the reprogramming occurs. So the particle itself has an activity, and that activity is delivering a tolerogenic signal to antigen presenting cells and carrying the cargo that gives the specificity to the antigens that are encapsulated.
And is this expected to be one and done therapies, or will patients need to be occasionally redosed?
In all of our data to date, from the animal models, it is a one and done. But from a human perspective, and to bring products to market and bring them through an efficient regulatory process, we're going to go with quarterly boosters. And there's an argument that there's a T regulatory system that when you reintroduce the antigen, you actually are enhancing T regs, and you're allowing the body to keep remembering that these antigens are healthy and they should not be attacked. So it's going to be a quarterly booster system. Over time, it may be a one and done, but I think that would take several years to determine. When we do through human studies, we find out how long is the durability of effect.
COUR has partnered with Takeda on a nanoparticle treatment for celiac disease. For listeners not familiar with the condition, what is it?
It's an attack against the consumption of gluten, and it goes, T cells, actually, you can actually see them and you can biopsy them. They actually attack the small intestine, and it's not very fun. So people get nauseous. They cannot tolerate pizza, beer, anything that has gluten in it. And there is no cure for this right now. There is no therapy for it other than going on a gluten-free diet. And even then, there could be accidental consumption of gluten. And there's people that still, even under a gluten-free diet, they still may get a real intolerance to the gluten.
And this can actually have serious consequences. How does the condition manifest itself and progress?
Like John spoke earlier, essentially the T cells are activated. So this is a CD4 activation that starts this positive feedback loop where they're activated. They'll migrate to the gut. Once they're in the gut, essentially they'll start destroying the gut by activating CD8 and NK cells and starting an entire inflammatory cascade where then other immune cells become involved. They'll send out signaling mediators. And so then the villi are destroyed, and the villi are critical to gut function. And essentially the gut will no longer function, and then that results basically in all of the reported symptoms that you see or hear about. But like John also mentioned, you take the way that people are diagnosed with having celiac disease. Is there the current therapy, if you will, is for people to be put on gluten-free diets, and then they'll take a biopsy of their gut, and then they'll look for healthy villi after they've been on a gluten-free diet for some amount of time. And then once they take the forced consumption of gluten, which actually causes pain to the patients, and then take a secondary biopsy, and then they can actually you see pictures where the villi are shortened, showing that there's actual destruction of the gut.
But what are those symptoms, and what damage occurs to the body over time with celiac disease?
Well, I'm going to, Adam, I'll let you add after me, but let's envision a forest with robust trees and greenery and vegetation and everything you would expect in a forest. And take a look at your gut after you have some serious, maybe 10, 20 years of celiac disease, and it's like a deforestation of that forest. So the trees are less, the greenery is less, some of the trees are down, and so your body, your gut that needs that robust forest preserve, if you will, to be able to absorb nutrients, to be able to digest things, breaks down. And so you can become malnourished. You can have significant gut ache continuously. Diarrhea is a very, people who have this, 30 minutes later, they're having events, needing, and they're in a lot of pain. So they're in a continuous pain mode, and they just have a dysfunctional gut that really doesn't recover. It just gets worse and worse over time.
As long as you're exposed to gluten, I was going to say that's a differentiator from other autoimmune diseases in the sense that in celiac, you can remove the antigen that's driving progression of disease. And once you remove it, so when patients go on gluten-free diets, their guts can basically recover because the gut is one of the few organs that can basically recover and the cells can go back to normal. But if you take something like type 1 diabetes or myasthenia gravis or PBC, those antigens cannot be removed. They're always there. And because of that, those patients do have continual progression, or MS. So in all of those situations, the antigens are inside of us. And so there's constant exposure to the antigen. So those patients will continually progress. Whereas celiac is unique in the sense where you know what the driving antigen is, and it comes from eating gluten. You can remove that, and in that way, you can stop progression of autoimmunity in those patients. So it's slightly different than other autoimmune diseases in that sense.
But Adam, wouldn't you argue that's a highly controlled lifestyle.
Right. I mean, and that's one of the reasons that Takeda looks at it as an open field where there's still a lot of people where there's unmet need because of what it's required for those patients in order to avoid gluten to that extent, because some people are extremely sensitive and even just a slight exposure causes extreme intestinal distress, such that, these people will avoid going out to eat at restaurants. They'll avoid going out to picnics with friends if there's even the potential that they can have exposure. So yes, it does cause a strain in lifestyle. And so you see the same thing in allergy, which is not an autoimmune type of disease, but with people with peanut allergy, where people go through extremes to kind of have a different lifestyle. And that I agree with you, John, it forces a different lifestyle upon a lot of people.
And other than adhering to a restrictive diet, are there treatment options available today?
No.
What's known about your experimental therapy, CNP 101, which is also known as TAK 101?
When we did our proof of concept study, we did it as COUR before Takeda took over the program, and we were able to shut down the T cell response, and people were able to ingest actually large amounts of gluten over a 2-week period. And so when we looked at the T cells, we actually saw in humans a reduction in those dangerous T cells who took our therapy versus the placebo group who still had the inflammation. And if you look at how people are diagnosed with celiac disease, they have to go through a gluten challenge. And doctors told us that if they looked at the data on our patients that were dosed with our product, they would not have been diagnosed to having celiac disease.
In the partnership with Takeda, what responsibilities does each company have in advancing the therapy?
Takeda is running the program to build out their gastroenterology pipeline. They have full responsibility. The money they invest is from their coffers, and we play more of an advisory role. So we meet once a quarter. Sometimes we meet more frequently to advise on regulatory issues. We talk about safety always. The product is very safe across the board. And then we'll just talk about their plan with the product, and we learn what they plan on doing, and then we go over timelines and items like that. So it's pretty much a Takeda-owned program, and we play an advisory role. There have been times we get asked questions because we are dealing in multiple disease areas, and we have a lot that we can teach them.
And what has the partnership meant in terms of validating COUR's approach?
Oh, I think it, in many respects, put us on the map, because there are a lot of companies that talk about tolerance, and they'll put some data out in a paper, they'll raise some money, but we actually did a proof of concept study in humans with positive results, and thus far, nobody has ever done that before. And so we feel we're at the forefront in immune tolerance, and we don't feel that the other technologies that are out there are doing well. In fact, we're seeing more failures occurring with those technologies, and we see us succeeding more in various disease areas.
It would seem that this is a technology that could be leveraged through partnerships. How do you think about balancing partnerships like the one with Takeda versus building your own pipeline of therapies?
It's a great question, Danny. We're always discussing. We have a call after this meeting today to discuss what are the, there's hundreds of autoimmune disease where there's absolutely no other therapy. And we're doing an assessment as we speak, and we continue to do assessments of what products are we going to bring forward. And then we also say, there's certain areas where there's a partner that comes to us and says, we've been in this space for 20 years and we think your technology is the best and we want to partner with you. So we're going to have a blend of proprietary programs that we own and then some partner programs. And it's a very broad technology platform. So who knows where we go in the future, but I think it'll be a continuum of buildup of our pipeline and also a buildup of collaborations and partnerships.
Your lead wholly owned program is CMP 106. This is in development to treat the rare autoimmune disease myasthenia gravis. What is myasthenia gravis and how does it manifest itself and progress?
I think I'll let Adam take this. His sister has it, and he probably could speak very personally about it.
Yeah, myasthenia gravis is an autoimmune disease that's targeted towards the acetylcholine receptor for about 85% to 90% of patients with myasthenia gravis. There's 10% that have the disease that's targeted towards a different antigen, but for the majority of patients, that acetylcholine receptor is blocked, and then essentially that's responsible for binding onto a signal called acetylcholine, and then that triggers muscle activity. And so what happens at the neuromuscular junction in these patients is that there's lack of signaling, and so essentially you lose muscle strength or muscle tone, and so patients over time, depending on how because there's different progressions, but some patients are less able to open their eyes. Some patients have more difficulty breathing, standing up, moving. And so essentially anything involved with muscle movement can be affected by myasthenia gravis. And so it can be completely debilitating. And to the point, I was just talking to my sister the other day. She was saying that even admissions into certain types of schools, they will basically not let you in if they think that you won't be able to handle the rigors of school. So she had to get a letter from a physician essentially stating that she could attend a school because that she was capable. So it can be that bad. So it's basically stopping the signaling at the neuromuscular junction because of targeted blockade to the acetylcholine receptor.
There's been some recent innovation around myasthenia gravis. What's the prognosis for someone with the condition today, and how well served are they by existing therapies?
So the long-term therapies that people are maintained on are essentially steroids. And so that's helped many patients. And so they take that with the side effects. And so one of the original goals was to try to find ways for steroid sparing. And then, one of the other things that came out maybe 10 or 20 years ago was IVIG, and that could basically put off symptoms, could be 6 months. And in that 6 months, you may not have to take as much steroids, so you can get rid of some of the pain from that. And then just in the last few years, there was a new therapy called anti-FcRns that essentially reduces all antibodies non-specifically. And then that has improved quality of life scores in those patients. The one that has been approved was only approved for one cycle. And so it's yet to be determined what happens when someone is on a prolonged treatment that removes essentially all antibodies, if there's going to be increased viral infections or any other kind of side effects when someone no longer has antibodies. But now, after the first approval of Vyvgart, the first FcRns, there's lots of other FcRns that are in the pipeline. And then there's also other methods for removing the antibodies, which include removing B cells. So now there are CAR T therapies that are out there that are trying to essentially kill the B cells that make antibodies that can target that acetylcholine receptor. But that improvement that you get with that one cycle of Vyvgart essentially moves the quality of life 2 to 3 points. But again, it was approved, in the last few years, so there's no data yet on the extended use of Vyvgart.
And keep that in mind that you have to keep, if you really want to relieve people's stresses, you have to continually dose. And as Adam said, we're not sure what that means because, again, you're immune suppressing somebody on a continuous basis. And we just don't think that's going to play out well over time.
What's your experimental therapy, CNP-106?
So CNP-106 is essentially, we have our platform, as we described earlier, which is the nanoparticle, which has this active function. And so that's our platform. And in each one of our therapies, what we change is the cargo. And so the cargo for the myasthenia gravis, we have encapsulated all of the reported T cell epitopes. And we've also encapsulated, we used basically a machine program that calculated from people's potential risk portfolio or risk profile to MG what potential antigens could be come up, and we also included those. And so essentially we have a suite of antigens that are specific to the acetylcholine receptor that are encapsulated inside of 106.
What's known about 106 from studies that have been done to date?
So we've done studies that have shown that we can reduce the acetylcholine receptor in the preclinical model and also improve the clinical outcome. And we've done all of this in the mouse model, but we're looking to extend this into the rat model to see something that is more closely related to the human. But we're also have our clinical trial open that will start treating patients, or not, we'll start the clinical trial where patients will receive the therapy, and then that'll happen in the next quarter.
What will be the development path forward?
I mean, the IND is currently open. So development path is essentially, patients will be administered, and then the primary endpoints for our first trials are always safety. So we need to show that we can safely administer the drug, but we also measure quality of life, and then antibody and T cells. So we look at both the immune and clinical parameters in these patients, and we have all of those parameters as exploratory. But for the first trials, we always look at safety as the first endpoint, and then we track the clinical and immunological endpoints such that we can power our next studies with the right amount of patients.
And we do look at safety, but we're also looking at clinical outcomes, and we're looking at T cells. And with that data, once we complete that study, we'll actually do a 2B study, which is a mini pivotal study. So we'll take what we learn from our POC study, and we'll then move into a pivotal trial. And then the next phase after that, depending on discussions with the FDA, we may do another repeat trial to get a label. So we have a pretty good model of how to bring these products into the clinic and then into the market.
The company raised $105 million in January. How far will existing cash take you?
Through 2026.
And is there plans for raising additional capital?
Yes. We're always talking to investors, and they're always taking a look at us. We may do another partnership, and I say may. We're always in discussions with large pharma about partnerships. And if the markets get a little better, of COUR is probably a very ideal candidate for an IPO.
John Puisis, President and CEO of COUR Pharmaceuticals, and Adam Elhofy, Vice President of Research for COUR. John, Adam, thanks so much for your time today.
Thanks, Danny.
Thank you.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.